Hi everyone, this is Mrs. Belong, and today we're going to talk about chromosomal abnormalities, or errors that can occur during meiosis that lead to the inappropriate number of chromosomes being in the gametes. Now, this can occur in a couple different ways. You can have something called a non-disjunction, and that's where you have your either your chromosome pairs not separating out appropriately in meiosis 1, the homologous chromosomes that are lined up right here. We can see in this cell down here, we have a cell in metaphase 1 of meiosis, and those homologous chromosomes should separate out appropriately to opposite sides. And if they don't do that, then you can have an issue, and I'll show you that in just a second. You can also have the sister chromatids not separating out appropriately in meiosis 2 as well. And any of those things are going to lead to too few chromosomes in some gametes and too many chromosomes in other gametes. And we don't want that. We want to have exactly the correct number of gametes or of chromosomes in our gametes. So here we have a cell in metaphase 1 of meiosis 1. You can see the homologous chromosomes are lined up right next to each other along that metaphase plate. Now, what should happen when the cell division occurs in anaphase, these chromosomes should be pulled to opposite poles, and we should see a separation, and we're generating gametes that are both haploid after we run through meiosis 2. As we can see here in the middle, we've got this would be what occurred after meiosis 1. We still have the sister chromatids attached. After we ran through meiosis 2, we would actually end up with 4 of these gametes here that are all haploid with 1 copy each of every chromosome. Now, what can happen in a non-disjunction is something like this. 2 of the chromosomes don't separate out, and so what that leads us to have are gametes that are going to have either too few chromosomes or too many chromosomes, as we can see here. With our one gamete here is missing a chromosome, so we list it as n-1. So the appropriate number of chromosomes -1. And this one right here has an extra one, so we've said this is n+1. So this could really occur in either meiosis 1 or meiosis 2. And as you can see, if the non-disjunction occurs in meiosis 1, all of the cells are going to have the inappropriate number of chromosomes, leading us to have all of our gametes having either a +1 or a -1 in terms of the amount of genetic material present in that gamete. Now, if we have if the non-disjunction occurs where sister chromatids fail to separate in meiosis 2, some of the gametes could be appropriate. Like here we have this normal separation in meiosis 1, but then when we get to meiosis 2, we have this chromosome failing to have the sister chromatids separate. So in this case, we end up with 2 of the gametes that have the inappropriate number and 2 of the gametes that have the appropriate number of chromosomes. And I feel like this is always an interesting question that is or could be asked on a test about when that non-disjunction occurs. So what's the issue with this? So what? We have extra chromosomes or we are missing chromosomes? Well, what occurs when a gamete that has too few or too many chromosomes is used to fertilize either an egg or a sperm, the baby is going to have the wrong chromosome number. And if they have an additional chromosome at any one given pair where they should have only 2, that is called a trisomy. So there's 3 copies of a chromosome instead of 2. And this is the example of a trisomy. Let's say that mom's egg has an additional chromosome here from a non-disjunction during meiosis, and dad's sperm has the appropriate number of chromosomes. And for this case, there are 3 chromosomes that we should have. And so we have a N and we have an N+1. When fertilization takes place, that baby is going to have 3 copies of that 3rd chromosome there, which is considered a trisomy. Now, you could also have something called a monosomy, where you're missing a chromosome or you only have 1 copy of that particular chromosome. And so here in this example, we have mom's egg missing that 3rd chromosome. Here's dad's sperm. And so mom's egg is an n-1. When fertilization takes place, now we only have 1 of those chromosomes, and that is considered a monosomy. So you have extra genetic material or missing genetic material. This should be okay, right? Well, for most of our chromosomes, deletions or additions are too disastrous. Our development is such a wonderful symphony of genes turning on and off at just the right moment. And additional genetic material or missing genetic material can really throw that off. Most trisomies or monosomies are too disastrous for the baby to even fully develop or be born. There are some that are survivable. And unfortunately, human chromosomal disorders are in a pretty high frequency in humans. Most embryos are lost to miscarriage because the alterations are really quite disastrous. It's a big issue with biochemical problems when you are looking at the development of a baby. It's a huge, and very intricate, very, very specific thing, the way that the genes are turned on and off and at the appropriate time. Now, there are some of the duplications or deletions that upset the balance a little bit less and are survivable. It would still leave the individual affected with a set of symptoms considered a syndrome. And one that you're probably familiar with or have heard of is trisomy 21 or Down syndrome. This is a extra copy of the 21st chromosome. Chromosome 21 is the smallest of the human chromosomes, so it has the fewest amount of genetic material. So when we look at the chromosomal abnormalities, or in this case, the instance of Down syndrome associated with the age of mom, we can see that it definitely increases over time. From anywhere under about 1 in 1,000 if you're under 30, and it does still happen in young mothers. It's just much less frequent than it does in mothers who are a little bit older. When you are 49 and have a baby, the chance of a chromosomal abnormality, specifically in this case here, Down syndrome, is about 1 in 12. So older the mom is, the more frequently we tend to get these errors of meiosis. There are a variety of different genetic tests that are available for mothers when they are pregnant. Right now, there's a lot of research being done, being able to look for markers in maternal blood. So mom just giving some blood and being able to look for markers for different abnormalities or genetic issues. But I do want to show you this is called an amniocentesis, and it is it looks terrifying, but you have a large syringe that goes into the mom's belly and removes some of the amniotic fluid that surrounds the baby. The fetal cells are then analyzed and a karyotype is generated. Now, this test allows doctors to look and see if the chromosomes are all appropriate. If they are broken, you can see if there's duplications or deletions, but you cannot test for any specific genes, just chromosomal abnormalities using this amniocentesis. Okay, so we talked about what can occur when you have a non-disjunction in meiosis and you have an extra or a missing chromosome and the impact that that can have. Well, errors of meiosis can also occur when you change the structure of the chromosome during prophase 1 when crossing over occurs. This crossing over, remember, the homologous chromosomes line up next to each other, they cross over, they break, and then they refuse together in order to mix up that genetic profile a little bit, and the gametes are really genetically unique. But when this crossing over, breaking, and refusing occurs, you can also have some issues. One is a deletion that can occur. A piece of a chromosome can not be included. A loss of a chromosomal segment would be very impactful because that segment itself is going to have a lot of genes that would then just be missing in that baby. Or you could have a duplication, which is a extra copy of a section or segment of the chromosome. So a segment is actually repeated. So you have that section of genes twice on the chromosome. Now, extra genes, as we, I think, already established, are not ideal. You could also have an inversion where a piece of the genetic material was actually reversed, and so flipped over and going in the wrong direction. Our enzymes that read our genetic material would not be able to read that if it was going in the wrong place. And then translocation is where one segment of a chromosome might be moved to a different chromosome. It can definitely, it's messy. All of our genetic material needs to be in the right place in order for the machinery inside of our cells to read it. We also are going to want to have the correct amount of genes. 2 copies of each gene, one from mom and one from dad on those homologous chromosomes. While the errors of meiosis lead to chromosomal abnormalities and a disorder or a syndrome within that particular individual who has been affected, it is not, again, considered an inherited genetic disorder. I hope you all have a wonderful day, and I'll see you in class.